The immune system is composed of two complementary responses: innate immunity and adaptive immunity. Innate immunity is the most ancient form of defense in our body. It is evolutionarily primitive and is pre-programmed to react to common broad categories of pathogens, and it kind of controls the infection and helps keep us alive, while our much more recent evolutionary invention, the adaptive immunity, gears up, adapts, evolves, and forms its army to better recognize this infection and eventually eliminates it, and even record and remember specifically each particular pathogen. Now what we'll be doing in this video is comparing the components and the characteristics of these two interconnected responses, and at the end fitting all the knowledge into a nice table that summarizes all the differences between these two systems. But before getting into all that, I want to ask you guys, if you like what we're doing and you find it helpful, please support us and hit the like button. Leave a comment and tell us your thoughts, and also share it with your friends. And we would really appreciate it if you subscribe to our channel. Now let's get started. Both the innate and the adaptive responses are composed of barriers, immune cells, serum proteins, and receptors. The barriers of the innate immune system include physical, chemical, and biological barriers, and also some other mechanical defenses. These barriers act as fixed defense mechanisms against infection and form our body's first line of defense. The physical barriers are formed mainly by the exposed epithelia of various tissues, the tight junctions that hold the cells together in those tissues, and the mucus that covers almost all barrier tissue surfaces except the skin. So a pathogen, in order to invade our body, must penetrate this tough physical barrier. It must manage to escape the highly viscous mucus layer, and then either dig through the cells or squeeze in between their tight junctions into the intercellular space. Now the second of these barriers is the chemical barrier, which consists of an arsenal of chemical substances that are lethal to foreign invaders. An example of such chemicals are the antimicrobial proteins and peptides. Antimicrobial proteins and peptides are specialized soluble substances that are mainly present in our body secretions or fluids, giving these fluids a unique cocktail of chemicals that are specific for each location. Antimicrobial proteins and peptides are very numerous and work by various mechanisms, but what they all have in common is that they constitute a chemical barrier against a wide variety of pathogens, mainly by weakening their effect, keeping them under control, and even directly killing them. The acidic pH is another example on a chemical barrier that acts against many pathogens. The acidic environment of some of our tissue surfaces makes it difficult for many pathogens to survive, because the low pH provides an inhibitory effect on microbial growth and also can kill pathogens directly. The next of our first-line innate barriers is the biological barrier, which is represented by our friendly microbiota that shares our body. These beneficial microbes are present on all barrier surfaces in enormous numbers, and they constitute an ecosystem that contributes to our defenses against infecting pathogens. As a biological barrier, the commensal microbiota prevent colonization by pathogens and makes it difficult for them to survive. Potential pathogens need to compete with our commensal friends for food and shelter. And on top of that, some of our commensals are actually armed with various weapons to deal with lots of pathogens. Finally, the mechanical and physiological defenses. Our body uses various defense strategies that mechanically remove the pathogens out of our system before they can manage to establish themselves. And these include mainly coughing, sneezing, lacrimation, urination, peristalsis, which may become diarrhea, and also vomiting, both of which we see a lot during many infections. We also have the shedding of skin and the movement of cilia. All these are mechanical mechanisms of pathogen elimination used by our immune system and have the same goal of expelling pathogens outside of the body. So these are the innate barriers that form our first line of defense and basically prevent infection by blocking pathogens from entering the body. And an infection can occur only if a pathogen is able to breach and surpass those barriers. Now, the barriers of the adaptive immunity are the secondary lymphoid organs, which include the lymph nodes, the spleen, and the mucosa-associated lymphoid tissue. These lymphoid structures act as barriers by filtering various antigens. The lymph nodes are specialized in filtering lymph and trapping antigens incoming from tissue spaces. The spleen filters blood and mounts immune responses to blood-borne antigens. The mucosa-associated lymphoid tissue filters the mucosal epithelium and initiates immune responses to antigens encountered along the mucosal surfaces. So these secondary lymphoid organs function mainly in trapping and filtering foreign antigens, and they provide sites for mature lymphocytes to interact with those antigens. And here are the sites where the transition from innate to adaptive immunity occurs. Now, the second component of these two responses are the immune cells. The innate immune cells consist of a wide group of non-specific leukocytes, including neutrophils, eosinophils, basophils, mast cells, monocytes, macrophages, natural killer cells, and dendritic cells. These innate cells are essential for our second line of defense against invading pathogens, and for that purpose they express recognition receptors that enables them to recognize certain patterns unique to some pathogens. And this recognition induces and triggers a variety of responses and defense mechanisms in these cells. Macrophages are the major phagocyte population resident in most normal tissues at homeostasis, and they are the first cells to encounter most pathogens. But they are soon reinforced by the recruitment of large numbers of neutrophils to sites of infection. These neutrophils have a great phagocytic activity, and they are the cells most immediately involved in innate immunity against infectious agents. Phagocytic cells have an important role in innate immunity because they can recognize, ingest, and destroy many pathogens without the aid of an adaptive immune response. Another group of cells that play a major role in innate immunity are the natural killer cells. Natural killer cells are the hitman of our immune system. They can detect transformed and viral infected cells and destroy them. And these cells are called natural killers because they can kill the target cells without pre-stimulation. Now, dendritic cells, although considered to be part of innate immunity, they actually function in making the connection between both the innate and the adaptive immune response. Dendritic cells can participate in innate functions and can ingest and break down microbes. but that's not their primary role in the immune defense. Their major role instead is to process ingested microbes in order to generate peptide antigens. Those peptide antigens are then carried to the surface of the cell and can be presented to T cells. And it is due to this process of antigen presentation that our lymphocytes will get activated and will be able to initiate an adaptive immune response. So these are the cells of the innate immune response. Now the cells of the adaptive immune response are the B and T lymphocytes. These B and T cells are activated upon antigen recognition. B cells differentiate into plasma cells to produce antibodies, which are essential for humoral immunity. T cells differentiate largely into helper and cytotoxic T cells, which are essential for cell-mediated immunity. Next, we'll move to the third component, which is the serum proteins. The serum proteins of the innate response include the complement proteins. Complement is a system of plasma proteins that complement the ability of antibodies and phagocytic cells to clear pathogens. These proteins act with one another in a cascade fashion to get activated. And once these complements are activated, they can serve as anaphylatoxins and initiate a local inflammatory response. Complements also can target pathogens both for lysis and for phagocytosis by cells of the innate immune system. Complement proteins can also come together and assemble to form a complex on the surface of some pathogens. And this assembly of complement fragments will create a large pore in the plasma membrane of the pathogen, which disrupts the osmotic integrity, leading to lysis and death of the targeted pathogen. This system of proteins, although considered part of the innate immunity, it actually cooperates with both the innate and the adaptive immune systems to eliminate pathogens, dying cells, and immune complexes from our body. Now, the soluble proteins of the adaptive response are the antibodies, or what are called immunoglobulins, and there are 5 main isotypes: IgM, IgD, IgG, IgA, and IgE. And as we mentioned earlier, our B cell progenitors will differentiate into plasma cells, and those plasma cells will produce very large amount of antibodies and will release them into the blood. Now, antibodies have 3 major roles in combating pathogens. Antibodies can neutralize and kill pathogens. Antibodies can also coat pathogens and target them for killing by our phagocytes. Antibodies can also activate the complement cascade. But keep in mind that unlike complements, an antibody attaches to an antigen only if that antigen matches the antibody exactly, like a key in the lock of the antibody. And that's why only the B cells that match the attacking pathogens are activated, so that only the exact antibodies that are needed will be produced. The final component is the receptors. Receptors of the innate immune system are referred to as pattern recognition receptors. These receptors are expressed by our innate immune cells and enables them to recognize certain patterns unique to some pathogens and dying cells. Pattern recognition receptors can recognize pathogen-associated molecular patterns, which are molecules that are shared by pathogens of the same type, and can recognize damage-associated molecular patterns, which are released from damaged, distressed, or dying host cells. These receptors are present intrinsically and are DNA encoded in the germline genes inherited from an individual's parents, and they are not generated through somatic recombination as the lymphocyte receptors. The innate receptors help rapidly recognize different sorts of pathogens, and that recognition induces and triggers a variety of responses and defense mechanisms in these cells against the invading pathogens. Now, the receptors of the adaptive immune system are variable antigen-specific receptors, and they include the B and T cell receptors. Our B and T lymphocytes possess those antigen recognition molecules on their surface, which allows them to respond specifically to the small molecular details of a foreign substance. And a very important word in that regard is the word antigen, which comes from antibody generator, because it can stimulate antibody generation. Antigen is defined as any molecule that can bind specifically to an antibody or to a B cell receptor, or can generate peptide fragments that are recognized by a T cell receptor. So an antigen could be any substance that can bind to a specific antibody or a lymphocyte receptor and can trigger and initiate an immune response. Now, B cell receptors, which are ultimately modified to be secreted antibodies, allow recognition of complete antigens, and they are capable of binding unprocessed antigens of virtually any chemical composition. Whereas the T cell receptors allow recognition of antigen fragments and are able to bind and recognize only peptides presented by specialized antigen-presenting cells. Both of these receptors are generated through gene segment rearrangements, so they are not inherited like the innate receptors, but rather arising from genetic recombination of receptor genes in each individual. So these are the main components of the innate and the adaptive immunity. And next we will move to the characteristics that shape those two arms of your immune system. The first two characteristics actually are related to the receptors that we just mentioned and what those receptors are able to recognize. The innate immune system is antigen independent and has limited specificity and limited diversity, while the adaptive immune system relies on antigens and is highly specific and highly diverse. So the innate immune system via pattern recognition receptors can recognize a small number of patterns on various pathogens due to its limited number of conserved germline encoded receptors. And that's what makes it limited in its diversity and specificity compared to the adaptive immune system, which can recognize over 1 billion specific sequences on pathogens, due to its highly specific and highly diverse antigen-specific receptors, which arise from genetic recombination of receptor genes in each individual. So the innate immune system is not very specific and not highly diverse, because it uses a limited number of secreted proteins and cell-associated receptors to detect infection and to distinguish between pathogens and host tissues. And although innate cells might be able to recognize gram-negative bacteria, they will not be able to distinguish the small differences among those gram-negative bacteria. For example, they can't distinguish between E. coli or Salmonella, but rather those innate immune cells are going to recognize a shared pattern between those bacteria. And such patterns are called pathogen-associated molecular patterns, and an example on these patterns is the lipopolysaccharides found in the outer membrane of gram-negative bacteria. So the pattern recognition receptors on your immune cells are able to recognize the pathogen-associated molecular patterns of the pathogens. The adaptive immunity, on the other hand, is very specific and highly diverse, because it uses a wide range of receptors that are able to distinguish the smallest differences between foreign antigens. So each B lymphocyte and each T lymphocyte is specific for a particular antigen, and they produce a specific response to a specific stimuli that target the pathogen specifically. And the adaptive immune cells not only knows that the pathogens are gram-negative like the innate system does, but also differentiates between the various types of bacteria and could tell which one is E. coli and which one is Salmonella. The adaptive immunity is so specific that it could even recognize the differences between various strains of these pathogens precisely. The next characteristic is the response time, which varies a lot between both systems. Innate immune defenses are very fast and are ready for immediate action, some occurring within seconds of a barrier breach, in contrast to adaptive immune responses which are slow and take days to arise following exposure to antigens. And the reason that the adaptive response is slow is due to the fact that the rare B or T cells that are specific for the invading pathogen and possess the perfect receptor for the job must first be found, and then must undergo clonal expansion over several days to allow the rare antigen-specific cells to increase in number sufficiently so that they can effectively combat the pathogen that elicited the response. So adaptive immunity relies on expansion of antigen-specific lymphocytes. And this expansion enables the body to have sufficient numbers of effector cells and antibodies that will eventually migrate to the site of infection to mount an effective immune response. Finally, the immunologic memory and response to repeated infection. Immunological memory is manifested as an enhanced ability to respond to pathogens that have previously been encountered and successfully eliminated. Innate defenses are not enhanced in activity upon subsequent exposure because it has no memory. So the performance of innate defenses is not improved by repeated stimulation and will not get better with time or exposure, and will respond in the same way to all pathogens and foreign substances. The adaptive immune defenses, on the other hand, are enhanced with each repeated exposure because it has immunologic memory. So the performance of the adaptive defenses, its responsiveness and its efficiency is improved by previous stimulation, and it gets better and better with time and repeated antigen exposure due to the formation of memory cells that provide long-lasting specific immunity. So this table actually summarizes all the differences between those interconnected systems. And the take-home message from it is that the innate immune response is a really important early response. It prevents lots of infections and quickly eliminates common invaders in most of the times. And a lot of infections are in fact eliminated by innate immune mechanisms alone. But if this innate response is unable to control the situation, then it will call for help from the more advanced and sophisticated adaptive immune system. And it helps keep us alive while the adaptive immune system is gearing up its highly specific army of cells and antibodies that can eliminate the infection. The adaptive immunity, on the other hand, has developed to give us the best possible defense against disease. It is highly specific in recognizing and targeting pathogens. It is highly diverse, so it protects us against the maximum number of potential pathogens. It provides us with the needed antibodies. And it has immunologic memory to protect us against recurrent challenges. Now, although it is convenient to put the immune system into two separate systems or responses and divide it into innate and adaptive, especially for ease of discussion and for test-taking purposes, But in reality, the fact is that both of these responses are highly integrated and operate cooperatively and don't act independently of one another. There's a considerable overlap of the cells and mechanisms involved in each of these arms of immunity, and there are many feedback loops that continuously cause stimulation from one to the other. And it is clear that they work together as a whole, and there is no clear cut between both of these systems. So the innate immune response helps the adaptive immunity, and the adaptive immune response also helps innate immunity. Activation of the innate immune response produces signals that are required to stimulate and direct the behavior of subsequent adaptive immune pathways. And the full development of the adaptive response is actually dependent on earlier innate pathways. For example, the innate immune cells process and present antigen to facilitate stimulation of specific T lymphocytes, and will subsequently release cytokines that are necessary for the activation of T lymphocytes, as well as triggering the differentiation of B lymphocytes into plasma cells and isotype switching. The adaptive immune response also aids and stimulates an efficient innate immune response. For example, T lymphocytes produce cytokines that enhance microbicidal activity of phagocytes. Antibodies secreted by plasma cells can aid in the destruction of pathogens through opsonization and can also activate complement to enhance phagocytosis and attract inflammatory cells. We reached the end of this video. Don't forget to check out the notes and the multiple choice questions. Thanks for watching and see you in the next video.